Hello everyone and welcome back to our channel. My name is Vedanth and I'm a student researcher at the BigDataBiology-Lab. Today we're diving into the ARG Norm package. We'll explore what ARG Norm is, why it is useful, and how to use it. ARG Norm is a tool to normalize antibiotic resistance genes by mapping them to the antibiotic resistance ontology provided by the CARD database. Right now, many tools exist for annotating antibiotic resistance genes in genomes and metagenomes, such as DPARG, arg's-OAP, Abricate, ResFinder, and AMR Finder Plus. However, each tool will have distinct output formats. Let's look at an example. The gene TET-A can be represented as TET-A, like that, or TET-A with a bracket, or all caps, TET-A, or even TET-A. So all of these refer to the same gene. For small-scale projects, a human user can quickly evaluate these outputs. However, for large-scale metagenomics projects, this becomes infeasible. What argNorm does is that it normalizes these different output vocabularies to the same ontology or the same vocabulary, which is the antibiotic resistance ontology, or the ARO. An ontology is a structured model that defines different types of entities and the relationships between those entities in a formal and organized manner. The antibiotic resistance ontology provided by the CARD database defines antibiotic resistance genes, mutations, mechanisms of resistance, phenotypes, antibiotics, and their molecular targets. The ARO gives antibiotic resistance genes unique identifiers, or ARO numbers. argNorm maps ARGs to these numbers. For example, TET-A will be mapped to ARO 3000165. All right, so let's go ahead and see how we can install and use argNorm on our system. Before we start off, it must be noted that Python is a prerequisite. And so if you don't have Python installed on your system yet, now would be the time to go ahead and install Python through the link in the description below. All the commands that we're going to use in this video will also be available down in the description, so you can go ahead and follow along as well. All right, so let's look at how we can install argNorm on our system. So go ahead and open up your terminal. So I've opened up my terminal over here, and we simply need to use the command pip install argNorm. So argNorm is a Python package, and so we can just use pip to install argNorm on our system. I've already installed argNorm, and so this happened pretty quickly. It may take some more time for you. With argNorm now installed on our system, we can go ahead and already start using argNorm commands. So the first command that we're going to take a look at is going to be argnorm-h. This will display a help message, and so we can see the help message right over here. And the help message is going to give us information on usage and positional arguments and all the options that are available for argnorm commands. So we can see the help commands, the help option, databases, modes, whether or not input files are harmonized. Don't worry, so we're going to take a look at all of these options in just a moment. So that is argnorm dash h. In fact, argnorm dash dash help will also give the same help message back. And so you can use these two commands if you'd like to take a look at the available options without necessarily having to go to the documentation. So with argnorm dash h out of the way, we can now start looking at different use cases of argnorm. I have a few example inputs over here in the examples folder. And I also have two other folders inside, harmonized and raw. The harmonized folder will just have ARG annotations that have been harmonized through the harmonization package. And raw files will just have ARG annotations that have been taken directly from the ARG annotation tools. So, for example, Abricate or AMR Finder Plus. Okay, so before going ahead and normalizing these files, let's quickly take a look at the general format of most argnorm commands. So most argnorm commands are going to look a little bit like this. You're going to say argnorm, and then the name of the tool that you used to perform ARG annotation. And so this could be Abricate or AMR Finder Plus or DeepArg or ResFinder. Then you will have to give the path of the input file. And so this will be dash I, and then the path to the original annotation. So just like that. And then the source or the name of the output file. So dash O, and then the name of the output file. So we can just call this annotation result.tsv, just like that. So this is what nearly all argnorm commands will look like. Of course, we'd also have different options such as harmonized, hamro, HAMRONIZED, harmonized or the mode, but we'll just take a look at them in just a moment. But this is the general form of argnorm commands. Okay. And so now what I'd like to do is normalize a couple of these example input data. Let's start off with argOAP, harmonized argOAP reads. And so just breaking down the naming here, the name of the tool that we've used to perform ARG annotation will be argOAP. The reference database is the SARG database, and the mode that we've used is the reads mode. The other possible mode will be ORFs, open reading frames, but we'll just get to that in a second. And so what I'd like to do is run argnorm for this file. Um, as you can see, this is how the input data looks like. We have the input sequence ID, we have the gene symbol and the gene name, and other data. And so what we'd like to see after performing normalization would be another column over here called ARO. And we'd like to see all of these ARGs mapped to their corresponding ARO numbers. So let us run argnorm on this then. So let's go ahead and open the terminal. And we're going to say argnorm. And the name of the tool that we're using is argOAP. So argSO. And then the mode that we're using is the reads mode. So we'd say dash dash mode, and then we'd provide reads over here. Then the path to the input file. So I'll just copy the relative path of this file and I'll just paste it over here. And then the name of the output file that we are going to store all the normalization results in. And so I'm just going to call this args oap.sar.reads.normalized.tsv. I'll also call this Because this is a harmonized file, I'll also add harmonized to the name. So reads.harmonized.normalized.tsv. And because this is harmonized, we also need to provide the harmonized option. So dash dash harmonized. And so let's go ahead and run this, and hopefully this will normalize this file. As we can see right over here, 100% ARGs mapped. So that tells us the normalization was successful. And right over here, we can see that this is the normalized file. We have the same sort of data as before performing normalization. And then right over here, let me make this a little bit more clear. Right over here, we have a column. And as we can see, every single gene, every single entry is mapped to its corresponding ARO number. And so that tells us the normalization to the ARO was successful. Also note that argNorm also adds these two other columns, which will be just a second. And then, ooh, there we go. So we have confers resistance to immediate drug class, as well as, oh, there we go. Uh, ah, let's just do this. And so argNorm will also add two other columns to the ARG annotation. And so this will be confers resistance to immediate drug class and overall category of drug class. I'll be covering what this means in the next section of this video for drug categorization. But just do note that these two columns are also added with the ARO column. Okay, so that is ARG's-OAP reads harmonized normalized. That was a bit long. Now I'll just put this in the harmonized folder right over here. Okay, so we have successfully normalized this input file. Now let's go ahead and normalize a raw input file maybe. Let's go to arg's-OAP, reads, but this time we'll do the raw file. So let's open up our terminal again. I'll just clear this up. And now all we have to say is argNorm, and then the name of the tool that we've used, which is arg's-OAP. Then the mode in this case is also going to be reads again. Just like that. We're just gonna provide the mode. And now we just require the input file. So I'll copy the path for this and I'll paste it right over there. And then I'd like to call the output file args-oap.sarg.reads.raw.normalized.tsv. So this is the normalized file for the raw data. In this case, this is not harmonized, so we don't need to provide the harmonized option. So we don't require harmonized here. And so we can go ahead and run this, and that will also normalize this file. As we can see, 100% ARGs mapped. Let's take a look at the normalized file. And just before that, this is how the raw file looks like. Um, and now, hopefully, when we take a look at the normalized file, we should see the ARO column with the ARO mapping. So let's take a look at that. So as we can see, I'll just make this. All right, there we go. And so we have the same data as before, just with the ARO column with all the genes being mapped to their corresponding ARO numbers. All right. So now that we've done this, I'll also put this in the raw folder right over here. Let's now maybe normalize a couple of abricate files as well. One thing to note is that the Abricate tool can do ARG annotation with different reference databases. As we can see, we have abricate.argonote.tsv, abricate.card.tsv, abricate.megares, and abricate.ncbi, and abricate.restfinder. And so Argonaut, Card, Megares, NCBI, and RESTfinder are all different databases, with Abricate being a common tool. And so in this case, while running argNorm commands, we would need to use the dash dash db option and specify which reference database is being used. So let's take a look at how that works. So let's open up our terminal again. Let's say argNorm. And in this case, let's go ahead and normalize abricate.restfinder.tsv. And so we'll say argnorm. In this case, we need to say we need to give the name of the tool, which will be abricate in this case, followed by the database option, so dash dash db. And in this case, the database will be restfinder. And then the path to the input file. So let's just do copy relative path. So we put the input file as well, and then the output file. And so I'm just going to call this abricate dot restfinder dot. And so this is a harmonized file. So I'm just going to put harmonized here as well. Dot normalized dot tsv. Let's do this. There we go. And because this is a harmonized file, we'll also need to say harmonized, just like that. And so the only change here is that we need to specify which database we're using as well. Let's go ahead and run this. And there we go. So we can see 97.2% are ARGs mapped. And so just before we take a look at the normalized file, this is what the file looks like before normalization. And so we can see input sequence ID, input file name, and then gene symbol, gene name. And so after normalization, we'd like to see another column right over here with ARO numbers. Let's take a look at the normalized file now. As we can see, we have the same sort of data as before: input sequence ID, input file name, gene symbol, and right over here, if I just scroll to the right, we can see, I'll do this. All right. And we can see the arrow column with each gene mapped to their corresponding ARO numbers. All right, so that was good. Let's also do another example. So I'll put this in the harmonized folder. Okay, let's do another example. Maybe this time we can take abricate.megares.tsv from the harmonized file folder again. This is what the file looks like. So we have input sequence ID, input file name, gene symbol, gene name, and some more data. And so after normalization, we'd like to see an ARO column with different ARO numbers. So let's go ahead and perform normalization on this. So let's open the terminal again. I'll just clear it. We'll then say arg norm. The name of the tool we're using, which is again abricate, then followed by the database that we're using, BB, and this time will be megares. Then we'll need to give the path for the input file. So I'm going to copy this relative path, put that right over there. And then let's give the name of the output file, which is going to be abricate.megares. And so again, this is another harmonized file. So I'm just going to say harmonized, and then we're going to normalize it. So dot normalize dot tsv. And because again it's a harmonized file, I'll have to give the harmonized option again. And so let's run this command. And as we can see, 100% ARG is mapped, so normalization was successful. Let's take a look at the normalized file. And if I go over here, we can see the ARO column with different ARO numbers. And so that tells us that the normalization to the ARO was successful. We also again have the confers resistance to drug class and overall category of drug classes columns as well. We'll just get to that in a second. Okay, let us now maybe normalize a few other raw files as well. So before that, I'll just put this in the harmonized folder. Let us go ahead and normalize abricate.argNorm. And so let's say argNorm. And so just do note that this is a raw file, so we don't need to say dash dash harmonized in the options. So, okay, let's go ahead and write the command argNorm. The tool again is going to be Abricate. The reference database is argNorm. DB argNorm. And then let's give the path of the input file. Let's go ahead, copy that, put that over there. And then the name of the output file. So that'll be abricate.argonaut.raw.normalized.tsv. And we don't need to say harmonized. And so this should go ahead and do the normalization for the raw file. So let's go ahead and do that. Okay, and so here is the input data. So we have the file name, the sequence, start, end, and so on. And what we'd now like to see is ARO with these two genes being mapped as well. Let's go ahead and take a look at that. So we have this sequence here. This sequence would then be mapped to these two ARO numbers. Okay, very good. And let's now normalize one more file as our last example. Let's go ahead and do this. I'll push this into that folder, clear the terminal again. And now let us go ahead and normalize. Maybe we'll do another example with ORFs. And so let's go ahead and normalize restfinder.restfinder.orfs. And so the mode that we saw previously was the reads mode. So we did normalization for this. So let's now go ahead and do normalization with the ORFs mode as well. And so let's go ahead and type out the command argNorm. In this case, the name of the tool will be RestFinder. Another thing is RestFinder, both of them are using the same database, RestFinder again. And so we don't really need to specify DB RestFinder. We don't need to say this. argNorm will do that automatically for us. So let's go ahead and give the mode. In this case, the mode will be ORFs. Then the path to the input. So let's go ahead and copy the relative path for that, paste it over here. And then I'm just going to call the output. Well, let's just go ahead and call the output restfinder.restfinder. And so this is a raw file. We're just going to call it raw.normalized.tsv. And we should be ready to go. So let's go ahead and run this command as well. As you can see, 100% ARG is mapped. We should again see the same sort of things happening. We should see an ARO column uh, with all of these genes mapped to an ARO number. Let's go ahead and see how that looks like. And let's do this. And there we go. So we have all the genes and the ARO column with all of these genes being mapped to an ARO number. Besides performing normalization, argNorm also provides drug categorization of drugs that antibiotic resistance genes confer resistance to. For example, the PBP2B gene confers resistance to the drug classes penam, cephalosporin, and cefamycin. These drug classes can then be categorized into a broader category of beta lactams. argNorm provides support for this classification and adds the confers resistance to immediate drug class and overall category of drug class columns to ARG annotations. The confers resistance to immediate drug class column will contain entries with all the drug classes that a gene provides resistance to. So that would be penam, cephalosporin, and cefamycin in the previous example with their corresponding ARO numbers. The overall category of drug class column will contain entries of the broader categories of the drug classes in the confers resistance to immediate drug class column. So that would be beta lactam in the previous example with its corresponding ARO number.